Hello, and welcome to UmojaHack Africa 2021.
My name is Nicolás López Carranza. I am InstaDeep DeepChain lead, and I will explain a bit of how this challenge works. The whole idea of this challenge is to create a neural network that is capable of evaluating neutralizing antibodies for the next influenza pandemics. And we are going to do that with the help of a DeepChain.bio platform and some data generated with the platform that you guys will use as training data for your evaluator. First of all, I would like to walk through the virus and how is the life cycle of the influenza virus. The virus has a very similar shape to the coronavirus that we all have seen recently in the news. The difference is that the coronavirus has a spike protein, and this virus has an HA protein in the surface of the cell. This protein gets attached to the sialic acid in the host cell receptor, and that's how the virus gets into the cell and injects its RNA so it can create all the proteins that form the virus that then they surface in the surface of the cell and generate a new copy of the virus. So the virus is hijacking the protein production mechanism of the cell to create more copies of it. We're going to try to stop doing that. By blocking the entrance of the virus into the cell. And the blocker will be our antibody designed and evaluated with our own network. How cool is that? So let's move to the notebook, and this is the notebook you guys have access to. The notebook, as I mentioned, is going to explain how to create a neural network that is able to estimate binding energy between the antibody and an influenza HA protein. So how this works? Well, here you have a very simple schema from what I showed before. You have the influenza virus that accesses the cell through the cell receptor here. But what if we create a neutralizing antibody that is a very good binder to the hemagglutinin HA protein, so hence the virus cannot get into the cell. So this is our task, to be able to create a method to evaluate how good is a given antibody in binding to the hemagglutinin receptor binding domain. So how are we going to create a scorer for an antibody binding? Well, this is where DeepChain gets into play. Because the first thing we can do is we can take a 3D structure of the antibody and the receptor binding domain of the HA protein and visualize it on DeepChain. And this is what I have here. I have loaded the file, which is, by the way, available for you guys to load, and you can create a free user on DeepChain and play around with it. Where we see clearly a few chains. We have the receptor binding domain of the HA protein, and we have the two chains that form an antibody. What we see is that there is some amino acids in the chain that are in contact or very close to the receptor binding domain of the virus. On DeepChain, we can identify very quickly that just by simply selecting the two interfaces, which, for example, let's select here, let's build an interface between the chains A and B, okay? And then let's paint in blue the chain C. So we have two different parts, and this defines hence a unique interface for this structure. And we can auto-select the positions that are very close to the chain C. And here we see these positions here represented on the antibodies sequence. You will notice that there are three parts of the sequence that are in contact with the receptor binding domain. Okay, with the RBD of the virus. These three parts are what we call in an antibody the complementary determined regions, which are called the CDRs, and you see them here again in this plot from Wikipedia. So these are part of the variable chains of the antibodies, and they are the ones that we are going to be evaluating. Because if we wanted to evaluate the whole sequence of the antibody, we would really struggle to generate a neural network that can learn out of such a long sequence. I don't say it's not possible. We are giving you the entire sequence, and you can build machine learning systems with the whole sequence. But the example of today is going to be focused on three small inputs into the neural network, which are the three CDRs. So once we downloaded all the data, which is available in a Google bucket, I uploaded it into a Google bucket for you. It is a public Google bucket, so you just can execute these lines and you will get the training data here. you can visualize this data. So we are going to load it into the data frame, and here you see which are the data columns. An ID, which is a unique ID per sequence, the whole sequence of the antibody chain, which is essentially 221 amino acids long, and then the 3 CDR regions that I visualize here on DeepChain. CDR1 is here. We are including the L as well, CDR2 and CDR3. So these are the 3 regions of the antibody. So going back to here, and the label, so basically, which is the binding energy per sequence. So essentially what we did is we took DeepChain and we modeled this antibody and we calculated the binding energy, and we generated this for a lot of different antibodies. So we mutated the antibody and we evaluated the binding energy, and hence we generated sufficient data for you guys to create a binding energy estimator. Once we have the training data, what we are going to do is split it between training and validation sets, so we can use them for our training. And we will define a batch size. Here I fixed it in 512. The input is a sequence of amino acids of length 5 here, then length 1, 2, 3, 4, 5, 6, 7 here. So 3 sequences of amino acids, and the output is going to be binding energy. These amino acids, they can be 20. If you see the introductory video, you will learn more about the biology. But here we create a vocabulary set for the amino acids, and we turn it into some sort of a tokenizer. So basically, we turn it into an integer. The next thing we are going to do in this part of the script is map the amino acids to integers. So take all these 3 columns and convert them in a list of integers. And here you have the result. So essentially now we converted these tokens into integers. And still we have the binding column. Here we are going to create the dictionaries for the inputs of our models, which they accept NumPy arrays. So we are basically converting this into NumPy arrays, and we do the same for the train, validation, and test sets. Here I'm showing an example of how the validation looks now, like a NumPy array. And CDR1, CDR2, and CDR3 are the 3 columns, which are essentially, again, these 3 CDR regions. So here we have the function to prepare the model. For this, we are going to be using Keras API from TensorFlow. We see a few layers here, and I also created a picture of how the model looks like, so you guys can follow a bit. But essentially, we have 3 input layers, and then we will have an embedding layer, which will be concatenated here. And we feed this into a bidirectional LSTM. And you could play a bit with the number of units or number of layers. But I also suggest that you explore a bit the transformer architecture in this problem to get maybe a better score. And after the bidirectional LSTM, we added a few dense layers. I mean, there is no dropout here, so I think this could be commented out. And finally, an output layer, which essentially has a single output, which is a binder. Here we are going to be applying the embeddings and generating the model. We are using Adam Optimizer, and we are compiling the model here, taking the optimizer, the loss, defining the loss, the optimizer, and compiling the model. We are printing the model summary here, which is essentially this, and it's for us a 3 million parameter model. Here we are just plotting the model. So after that, we are training the model. Here I did only 5 pokes because I'm on my laptop, and you see here the model, you see it learning at so the loss is decreasing a bit, but I guess we need to basically train it for a bit more of a box. I will let you guys do this. And once we have our model trained, we can run model.predict and generate a prediction out of the model, and we can generate a score CSV. So once we have the binding, which is the result of our model predict, we can generate a submission. So we are going to take the sub.csv sub, or for example, it has this shape, like kind of sample. And we are going to upload it to the UmojaHack page. So essentially, for those who never worked on, there's going to be a place here where you can upload the file. And that's all about it. I think that this is first a good start on how to generate a first model And so any questions, we will be available during the hackathon to explain. And thanks for joining.